Welcome to our virtual tour of alzdata.io, a new resource that's displaying highly unique human image data of Alzheimer's disease at never-before-seen microscopic levels. This website presents a rare case of highly preserved human biopsy data from a patient suffering from severe Alzheimer's-related dementia. Throughout this video, we're going to guide you on a tour of the main pages of the website. We'll start by diving into the background of the project, covering the initial discoveries and the inspirations that led to this work, and then we'll explain what kind of data was collected, how it was analyzed, and what insights we've gained from it. After that, we'll introduce some of the interactive tools that were developed to help you explore these datasets in more depth. Finally, we'll showcase several of the datasets themselves, taking a closer look at the scientific context behind the data and highlighting some of the key features you can explore here. This website offers a detailed overview of the remarkable collection of legacy biopsy brain samples from Alzheimer's disease AD patients. These samples, originally collected by prominent AD researchers from the 1960s through the 1980s, are incredibly rare and valuable. What makes them so unique is the exceptional preservation of their ultrastructure, which has been maintained throughout the decades, as frozen resin samples do not degrade. This level of preservation allows us to examine intricate cellular features that were previously inaccessible to researchers. And now, with the use of new deep learning-based analysis techniques and advanced microscopes with much higher resolution, we can explore these biopsy samples in unprecedented detail. Let's start with the background page. This is the homepage of the Alzheimer's 3D-EM database and provides an overview of this important project, beginning in 2020, and supported by the NIH's National Institute on Aging. Background information regarding the samples and initial experiments is also mentioned here. The database not only serves as a repository of image data, but also provides tools for further exploration and analysis, fostering collaboration among researchers. The page highlights the integration of modern imaging techniques, showing how advanced imaging methods and deep learning approaches are being used to analyze these historic samples. By offering open access to this wealth of information and innovative tools, the Alzheimer's 3D-EM database aims to drive future research and deepen our understanding of the complex mechanisms underlying Alzheimer's disease. Moving to our Discovery page, you'll find comprehensive information about the cellular features and discoveries that motivated the creation of this database. Modern research focuses primarily on discovering the causes of early-onset Alzheimer's disease cases. However, at the time these biopsy samples were collected in the 1960s, basic characteristics of Alzheimer's dementia were not known, and the figures displayed on this page were some of the clearest new features when the tissue was first analyzed. Since these first analyses, and further augmented by new analyses on this website, many more biological features of Alzheimer's disease have been characterized and rigorously studied. Now, let's dive into one of our key features, the Analysis page. This section connects you to a variety of web pages that showcase the results and datasets derived from our extensive image data. We've employed cutting-edge techniques, including serial block face scanning, electron microscopy, and advanced deep learning approaches, to extract detailed information about inter-organelle associations and the reorganization of the cytoskeleton linked to neuronal degeneration in Alzheimer's disease. These methods allow us to gain deeper insights into the cellular changes that occur as the disease progresses. Labeling 3D image data is a labor-intensive process, so we have leveraged deep learning tools to automate much of this work, significantly increasing efficiency. This automation relies on the availability of training data, which is crucial for developing effective algorithms. You can read more about the specifics of this training data we created in the first section here. One of the powerful auto-segmentation tools we utilize is CDeep3M, an open-source deep learning tool specifically designed for large-scale image segmentation tasks. By clicking the CDeep3M link, you can access the research article we published that details our findings and methodologies. Once a deep learning workflow has been successfully applied, we are able to produce rich data reconstructions that reveal intricate cellular details, with an example provided in the reconstruction data section. Additionally, we offer further sections that display what the deep learning inference data looks like and how this data can be quantitatively measured. You'll also find links to computational workflows that can assist researchers in navigating and utilizing this valuable information for their own studies. Moving on the collected data page. We have collected an extensive array of image data and offer several different ways to search for and explore it. The first two tabs, Hierarchy View and Image View, provide two distinct indexed methods for navigating the datasets, while the Subvolumes tab takes you to a specialized 3D image stack viewing tool for more in-depth exploration. Each of these options gives users flexibility in how they access and interact with the data. In the Hierarchy Viewer, the first level of the tree branches represents the neuropathologist's name, serving as an index for a specific biopsy sample collected or provided by that particular neurologist. The legacy samples in our database come from renowned neuropathologists, doctors Robert Terry and Suzanne Mira, who collected them in the 1960s and 1980s, respectively. These historical samples serve as a critical foundation for current research into Alzheimer's disease. The second level of the hierarchy organizes the data by biopsy block sample label. A biopsy block is a tissue specimen obtained through a medical procedure in which a small sample of tissue is extracted for examination. After the tissue is removed, it is processed into a block to facilitate detailed analysis. The final level of the hierarchy presents the image data itself, which is collected for various types of analysis. Initially, a 2D survey image provides a broad overview of the specimen, allowing researchers to evaluate its condition and identify specific areas of interest for further study. Within a three-dimensional volume, a region of interest, ROI, refers to a particular subset of data that researchers zoom in on for more detailed investigation. Some of our 3D ROI volumes highlight critical Alzheimer's disease features, such as paired helical filaments, PHF, and lipofuscin, allowing for targeted analysis of these key markers. Finally, by clicking the image icon at the last level of the tree, you will be taken directly to the detailed image page, allowing for interactive viewing of the images. Moving on to the derived datasets section, you'll find the results generated by our deep learning tools, including detailed 3D reconstructions and quantitative analyses of cellular structures. These datasets, powered by deep learning-based computer vision, showcase applications to segment and analyze key cellular components such as mitochondria, endoplasmic reticulum, synapses, and Alzheimer's disease-related pathological structures, including paired helical filaments, amyloid beta clusters, and dystrophic axons. Some of these datasets overlay 3D auto-segmentation predictions onto the original images, and others are 3D mesh-based visualization of structures of interest at nanometer and micrometer scale. For these datasets, clicking the 3D object icon will launch our 3D rendering tool for interactive visualization. Now we will move on to exploring how to use the interactive tools that were developed for this website and its datasets. First, let's start with the Image Viewer, which can be launched by clicking on any of the block image datasets available on the Collected Data page. Once you've selected a dataset, clicking the Detailed Viewer button opens our customized image viewer, designed for interactive exploration of complex 3D datasets. This tool allows you to seamlessly navigate through cellular layers, zoom in on specific organelles, and compare healthy cells with those affected by Alzheimer's disease. The viewer provides intuitive controls for easy manipulation of the dataset, making it an ideal tool for detailed examination. One of the key features of the image viewer is the Z-Slice slider, which allows you to control the current image plane along the Z-axis of the three-dimensional image cube. You can explore these slices by either sliding the Z-Slice bar or using the arrows above the viewer, which increment the current slice by 1 in either direction. For enhanced visibility, brightness and contrast sliders are also available, enabling you to adjust the view to highlight specific structures of interest at any level of magnification. In addition, the viewer offers bookmarking tools, which allow you to place pins or draw square or polygon-based boundaries around areas of interest. These bookmarks generate unique URLs that can be shared with other users of the database for collaborative exploration. To download these image datasets, navigate to the Image Data Download Options section found on the dataset's description page. This is the same location where the Detailed Viewer button was accessed. Each image we produce is carefully stored with detailed metadata, which is essential for comparing results across various experiments, subject preparations, imaging techniques, and biopsy sources. The metadata ensures that the context of each image is fully documented, making it easier to understand the nuances of each dataset. Additionally, a Digital Object Identifier, DOI, is generated for every image, providing a unique and permanent reference that ensures the data can be easily cited and accessed. More importantly, all of our data is securely protected by the UCSD Digital Access Management System and preserved in the long-term digital archive, Chronopolis, ensuring its integrity and future accessibility. The next interactive tool we developed is a viewer for 3D reconstructions of segmented data. This can be found by clicking the 3D render datasets in the Derived Data page. You are currently viewing the 3D rendering of an astrocyte cell process. The green mesh represents the astrocyte's plasma membrane boundary, while the yellow spheres indicate subcellular glycogen deposits. The blue structure corresponds to paired helical filament PHF array number 1, and the orange represents paired helical filaments. This 3D rendering tool allows easy zooming in and out using the mouse wheel, and you can rotate the object by holding the left mouse button and dragging left or right. Additionally, you can toggle the display of cell components in the 3D viewer using the checkboxes on the left side. alzdata.io enables you to explore a wealth of information regarding cellular changes associated with Alzheimer's disease, particularly by observing cells affected by the disease at various stages of severity. These affected cells are often situated adjacent to healthy ones, creating a unique opportunity to study the contrasts and similarities between them. This juxtaposition provides invaluable insights into the disease's progression and the intricate alterations that occur over time, enhancing our understanding of how Alzheimer's impacts brain cells. Within the database, you will find displays of paired helical filaments, PHFS, which are abnormal twisted protein strands primarily composed of hyperphosphorylated tau proteins. PHFs are a hallmark of neurodegenerative diseases like Alzheimer's, and their structural arrangement plays a critical role in the pathology of the disease. Alongside this, the database also features significant information about amyloid accumulations. Amyloid accumulations are formed by the aggregation of amyloid beta peptides, leading to the formation of plaques that disrupt cellular communication and contribute to neuroinflammation. These plaques are frequently found in the brains of individuals with Alzheimer's disease and are believed to play a significant role in the disease's pathophysiology. Together with paired helical filaments, these two features, amyloid deposits and PHFs, are considered the hallmark characteristics of Alzheimer's disease, highlighting their critical importance in understanding the condition and its progression. Additionally, we delve into modifications to subcellular organelles and the cytoskeleton, which are crucial in understanding the cellular dynamics in Alzheimer's disease. Notably, we examine axonal dystrophy, a phenomenon where axons undergo structural degeneration, resulting in impaired neuronal communication. This deterioration is pivotal in understanding how neuronal connections are disrupted, leading to cognitive decline. Autosomal degeneration is another critical aspect highlighted in our database. This condition is characterized by changes in the cell body that can lead to reduced neuronal function and survival. By analyzing these degenerative processes, researchers gain deeper insights into the overall health of neurons and how these changes contribute to the onset and progression of Alzheimer's disease. In addition to these features, we also explore the accumulation of lipofuscin, a complex age-related pigment that gathers in various tissues, particularly in senescing cells such as aging neurons. The presence of lipofuscin may indicate cellular stress and is associated with neurodegenerative diseases, including Alzheimer's and Parkinson's. Together, These observations enhance our understanding of the mechanisms underlying Alzheimer's disease, illustrating how both healthy and compromised cells interact within the brain. For more comprehensive information about our project and the resources we offer, we invite you to visit our About Us section, where you can explore detailed descriptions of our team. This team delved into the intricate aspects of the datasets and applied, innovated, and developed many of the techniques described on this website, such as serial block face scanning, electron microscopy, and deep learning approaches for image analysis. Additionally, you can explore CIL.org, which features even more examples of biological image data that can enhance your understanding of cellular structures and their implications in various diseases. At alzdata.io, we are dedicated to providing researchers and the broader community with valuable resources to address Alzheimer's disease. Our database not only highlights the critical features of the disease, such as amyloid accumulations and paired helical filaments, but also allows users to interactively explore the data through our advanced image viewer. We believe that this resource can significantly aid in your research efforts and contribute to the larger goal of understanding and ultimately combating Alzheimer's disease. We would also like to take this opportunity to acknowledge the contributors who have played pivotal roles in this project. Dr. Mark Ellisman, our principal investigator, along with Stephen Peltier, the executive director, have been instrumental in guiding the development of this platform. Willie Wong and Matthew Maddeney worked to build the website and various software tools and analyzes. Research scientist Sebastian Fahn has provided critical high-resolution tomography data. Lastly, Sean Pentecoff, our IT manager, has ensured that our systems run smoothly, allowing us to share this transformative tool with you. Additional histologists and contributors of datasets can be found by clicking the Contributors button at the top of the website. Thank you for joining us on this tour of a groundbreaking resource in neuroscience research. We hope you find alzdata.io to be an essential part of your journey in exploring the complexities of Alzheimer's disease and its effects on the brain. These new databases and your engagement promote the uncovering of new knowledge and advances in the field. Goodbye.